Okay, so before we get down to the biochemistry of what actually happens on the electron transport chain, let's first just have a little recap about the structure of the mitochondria. Okay, so you can see here the whole inner membrane of the mitochondria is covered with these tiny stalked particles, which we know are called ATP synthase. And obviously huge surface area for that. We've also remind ourselves of what we've got in the matrix, so that's the 10 NADH or reduced NAD, 2 from glycolysis, 2 from the link reaction, 6 from the Krebs cycle, and we've also got our 2 lots of reduced FAD, again another type of coenzyme that came from the Krebs cycle. So they're available in the matrix of the mitochondria and are going to be, I'll show you on the next slide how they're involved in the electron transport chain. So again, before we get started on the fine detail, just as a little recap of what this actually shows, and you can see here over on the left hand side our ATP synthase, sorry, on the right hand side our ATP synthase, so that's the stalked particles referred to on the previous slide. We know about chemoosmosis and how that's used, the flow of protons through that drive the synthesis of ATP. I'll show you that in a second. We've also got 3 proton pumps spanning the membrane. Okay, we're going to call those A, B and C. But you can also see there's some an extra level of detail on here. Actually these are complexes known as complex 1, but you've also got complex 2 here, complex 3 and complex 4. It's just that complex 2 is not actually a proton pump, it's just part of the electron transport chain. You can also again see an extra detail on here with coenzyme Q there in the middle, and at the top cytochrome C. They're just all part of the electron transport chain, but we're interested in these proton pumps, so we've labelled those as carrier A, B and C, and that's shown like that in the AQA textbook. It's also important to be aware of the orientation of this membrane. So at the top we're talking about the intermembrane space. Okay, so that's going to be between the outer and inner membranes of the mitochondria, and of course the matrix down at the bottom. So that's sort of the orientation of this membrane. Okay, so we'll talk about NADH first. It comes in, it reduces the first carrier. Electrons released are used to pump a proton across the membrane into the intermembrane space. Okay, and obviously as that electron continues to flow along the chain, at each carrier it pumps a proton across. So you've effectively pumped across 3 protons and the electrons come out at the other end. Okay, those protons are free to flow through the ATP synthase. So now we've got protons moving back into the matrix, given the energy required to drive the synthesis of ATP from inorganic phosphate and ADP. Just to kind of balance this out a little bit, of course, then this is where our water comes from. So we've got a half an oxygen molecule combined with a couple of protons, the 2 electrons that came from the carriers, and that's enough to synthesize our water. So just to consider the theoretically, NADH, it's easy to imagine that it actually provides enough energy, enough redox potential to actually generate 3 ATP molecules. This is theoretical. In practice that doesn't actually work. 3 protons pumped across, as you can see, 3 protons flow back through, enough to generate 3 ATPs. Okay, because we had 10 NADHs in the first instance, theoretically the NADHs provide enough energy or reducing power to the synthesis of 30 ATP molecules, 3 from each of our NADHs. So it's easy to kind of get your head around that. There are 3 proton pumps, 3 protons are pumped, 3 protons flow back through, 3 for each NAD. There are 10 NADs, so you can make 30 ATPs. As I mentioned though, that is theoretical. I will go on, probably I'll put it on a separate video to sort of talk for those of you who are really interested in doing biology or biochemistry at university and are more interested in this, just to have a little look at the actual situation, because of course that's theoretically the case and that isn't what actually happens, but that is way above the specification, so please don't look at that if you're happy to just accept this. Okay, so now using the same membrane, but just obviously taking off the NAD so it's not confusing, we'll talk about FAD. So this one comes in on the second carrier, so it doesn't actually get involved with that first carrier. You can see the same process as before. It comes in, it oxidises the carrier, the electrons are moved along from B to C in the process, pumping protons across the membrane, and those protons can then flow back through the ATP synthase, and then we've got our electrons and protons in the matrix that are able to combine with water. At the same time the ATP has been synthesized, the electrons and the protons combine with oxygen to make our water molecule. And so you can see, hopefully, that the only real difference is that FAD comes in on the second carrier. It just has a slightly different redox potential to the NAD. That means that for every FAD we have, we can only make 2 ATPs, not 3. Again, that's theoretical, but it's easy to get your head around that, to just imagine, because it comes in on carrier 2, it can only pump 2 protons, not 3. Therefore, only 2 protons flow back through, therefore only 2 ATPs are made. And as we have 2 FADHs that came from Krebs, 2 times 2 is 4. So theoretically, we're able to make 4 from FADH, 30 from NADH. That gives us 34 ATPs from the electron transport chain. And here's just another way of showing that, linking back to the very first slide, so you can see how how chemiosmosis works and how the flow of protons through the synthase generates our ATPs. Just a little summary now here over on the left, just so you can see theoretically where all the ATPs have come from. So as described on the previous slides, each NADH, and we have 10 of those, remember, can make 3 ATPs. Imagine 1 proton for each pump, and therefore 3 protons in total. 3 pumps, 3 protons, 3 ATPs. That gives you 30. The FADHs, they come in on the second carrier, so they can only pump across 2 protons, therefore 2 ATPs, and we have 2 FADHs, so that's the 4 ATPs, so that's 34 that come from the oxidative phosphorylation, or the electron transport chain. Remember, we had a net gain of 2 from glycolysis, and we also got 2 ATPs from Krebs. Add those together, that gives you 38 ATPs. So that's a number that you may well see quoted in A-level biology textbooks. Actually, it's not, they don't mention it at all in the newest version that you guys have got, probably because that's a theoretical amount, and in practice it doesn't work out like that, for lots of, as I said, lots of complicated reasons, which I won't go into here. Okay, so the last thing I wanted to talk to you about is an application style question where scientists have actually used inhibitors of enzymes in the electron transport chain. So you can see on here this table, inhibitor 1, 2 and 3. Actually the transfer of electrons along the chain does require require enzymes, and therefore if we can inhibit them, we can stop the transfer of electrons, and then we can look at the redox state of the carriers. We can look at whether they are reduced or oxidized when they've been inhibited. So you can see here they've just called them carriers A, B, C and D, and then inhibitors 1, 2 and 3. Interestingly, cyanide, which is a chemical I'm sure you've all heard of, a nasty poison, obviously kills people, that's actually what we call a respiratory poison. It actually inhibits cytochrome C oxidase. Not that you need to know that, but that's complex number 4, or on our diagrams, the 3rd proton pump. That actually also contains, it's a very complicated protein. It also contains the enzyme responsible for producing the water, transferring the electrons and hydrogens from cytochrome C onto oxygen to make water. So if you inhibit that one, the whole of respiration stops, the electron transport chain gets backed up. So that's why it's such a nasty poison. But anyway, back to this. So you would need to be able to basically work out the order of these 4 carriers. Which one comes first, second, third and fourth, and get those in order. Now, obviously I'm about to explain this to you, so by all means, pause the video and see if you can have a go yourself before I go on to tell you the answer. But I am now going to explain how to work this out. So we're looking for what happens when you inhibit something. So basically, if something's inhibited, then the electrons cannot be transferred any further along the chain. And so if you inhibit it, the carriers downstream would be oxidised. You can't give them electrons, you can't reduce them, so they remain oxidised. So if we look at carrier C, okay, you can see that it doesn't matter which inhibitor we put on carrier C, it can always be reduced. and therefore it has to be first in the chain. No inhibitors prevent electrons being transferred to it, so it has to be first. So now it's just a process of working through. So now we're looking for the next carrier. So if we look over at carrier A, we'll see that that's actually second in the chain. This is because two of the inhibitors, in the presence of them, it still is reduced, but actually we've got this inhibitor here. Inhibitor number 2 is able to prevent carrier A from being oxidised, so it must be lying in between carrier C and A, so it's stopping the transfer of electrons from carrier C to carrier A, so carrier A must be second. Okay, third in the chain, therefore, based on this, is carrier D. Okay, you can see that both inhibitors 1 and 2 are preventing the reduction of carrier D. It remains oxidised. Only inhibitor 3 has no effect, so it can be reduced in the presence of inhibitor 3. But inhibitors 1 and 2 are preventing its reduction. It remains oxidised because the electrons cannot be transferred to it because it's being inhibited. So that's carrier 3 in third position. And lastly, as you'd expect, carrier B. Okay, all 3 inhibitors prevent the transfer of oxygen, the transfer of electrons to carrier B. It remains oxidised in the presence of all the inhibitors. It can never be reduced, therefore it must be last in the chain. And actually, that, if you then put that into a real life context, carrier B therefore would be that final complex in the electron transport chain, tartrochrome C oxidase, the one that cyanide inhibits, and that's obviously puts a total stop to any respiration at all, so that's why it's lethal.